Hello everyone, welcome back. In the last episode, we explored the mysteries of electrophysiology. More specifically, we first talked about the resting membrane potential and then we learned about the action potential. These are the electrical properties of a single neuron. We can see how electrical signals go along within one neuron. Now, we're about to enter the world of communication. How does one neuron convey information to another neuron? That is related to synaptic transmission, which is the topic of this episode. In this episode, we will learn about the two types of synapses, electrical ones and chemical ones. Then we will go into details about chemical signals. We will learn about the neuromuscular junction as a prototypical chemical synapse. Then talk about neurotransmitter synthesis, excitation-secretion coupling, and then in the next part, we will see ligand-gated ion channels, G-protein coupled receptors, postsynaptic potentials and summation, reuptake and enzymatic degradation, and finally synaptic plasticity. In general, synapses are defined as places where two neurons communicate information. It could also be a place between the sensory receptor and a neuron, or between a neuron and an effector cell, for example, a muscle cell or a gland cell. There are two general types of synapses. They are electrical synapses and chemical synapses. This figure illustrates an electrical synapse between two neurons, and this one is a chemical synapse. Electrical synapses. Let's zoom in on them. Firstly, we can see that one characteristic of the electrical synapses is that the two neurons are very, very close. The gap between them is only about 3 nanometers across. It's so narrow that we may find it difficult even seeing it with an electron microscope. Electrosynapses allow the direct flow of ions through gap junction channels. The ions in the action potential of one neuron will flow directly to the second neuron through these channels. Each gap junction channel is made of 2 connexons, and each connexon is made of 6 connexins, as the zoom-in graph shows here. Electrical synapses allows a weakened synchronizing signal. That is to say, the action potential of the presynaptic neuron flows directly into the second neuron. We can see this clearly in the graph on the left side of the page. In the upper figure, we can see that the first cell has an action potential, in which the electrical potential goes positive, while in the bottom we can see the electrical potential of the second cell. The electrical postsynaptic potential, the electro PSP, only goes to -64 microvolts, which is only 1 microvolt above the resting membrane potential. Therefore, although the two signals are in synchrony, they happen almost at the same time. However, the electrical potential in the second cell only changes very slightly. If, however, there are many neurons that do this thing together, and there are many electrical synapses, then in the second cell, we will successfully trigger an action potential as well. This allows a high degree of synchronization between a group of cells. For example, in certain glands, we see gland cells that secrete things together. This is accomplished through electrical synapses. We can also see this in pacemakers. There are certain nuclei, or bunches of neuron, groups of neuron, that exist in the brain. These cells are in high synchrony. They help maintain the rhythm in the body, for example, the circadian rhythm, which is about sleep and awakeness. We can also see this in cardiac muscles. Cardiac muscles are branched muscles. They are usually Y-shaped and connect to various other cardiac muscle cells. Between these muscle cells, there are lots of gap junctions as shown here. Therefore, when one muscle contracts, then the calcium ion or other cations will flow to adjacent muscle cells, and they will contract together. That is why our heart can be so united and pump blood to all our body. Question time. At which of the following places do electrical synapses most likely exist in large quantities? A. In nerves, B. In the cerebral cortex, C. Sensory neurons in the skin, or D. Smooth muscles in the intestine walls. Try to think about the properties of the pacemaker and cardiac muscle cells that we just talked about. The answer is D, smooth muscles in the intestine walls. This is because in the peristalsis of the intestine, the smooth muscles need to contract together in order to push the food further down the intestine. That is why the smooth muscles have electrical synapses. In contrast, In nerves, there are no electrical synapses because nerves are the axon of neurons. They do not even have synapses. In the cerebral cortex, there actually do exist electrical synapses, but not in large quantities, because the cerebral cortex is where we process information. If every neuron is in synchrony, then the cerebral cortex will never be able to process information. And sensory neurons in the skin, they also don't have electrical synapses, because, for example, if we want to have touch, then we need to be able to distinguish between two stimuli that are very close to each other. If the sensory neurons fire together, if they're in synchrony, then we will not be able to distinguish between two stimuli. Now, it's finally time to come upon chemical synapses. Chemical synapses are much more useful than electrical synapses. They are very, very diverse, thanks to the various types of neurotransmitters and receptors, which we will talk about in a short while. Chemical synapses consist of three main components. They are the presynaptic neuron, the synaptic cleft, which separates the two neurons, and the postsynaptic neuron. In the presynaptic neuron, we see an axon terminal. This is the ending of an axon that connects with the second neuron. In the axon terminal, we can see many mitochondria because the axon terminal has to maintain a good ion concentration gradient. We can also see an active zone. This is something special to chemical synapses. The active zone looks very black in electron microscopes. That is because there are many vesicles that are there and many proteins, making this region seem very, very dense. There are synaptic vesicles. These are vesicles that contain neurotransmitters. Neurotransmitters are small chemical molecules or possibly protein peptides that are released into the synaptic cleft and drift across to the postsynaptic neuron. It binds to specific receptors on the postsynaptic neuron and triggers a response in the postsynaptic neuron. This is why chemical synapses are called chemical. The information is conveyed using neurotransmitters. We also have secretory granules. These look very, very similar to synaptic vesicles. However, secretory granules usually contain peptides, while synaptic vesicles usually contain small molecules as neurotransmitters. As a result, secretory granules look dark on electron micrographs, while synaptic vesicles are a little bit more light. In the synapse, we have the synaptic cleft, which is about 20 nanometers across. In the postsynaptic neuron, we see dendritic spines. These are little bulges on the dendrites of the neurons, and synapses are usually formed on these dendritic spines. There is also a postsynaptic density, just like the active zone in the presynaptic neuron. In the postsynaptic density, there is also a lot of protein, especially receptors and other proteins required for the response. Therefore, it also looks black in electron microscopes. And finally, we have receptors. These are transmembrane proteins. On one side of the membrane, they can bind to the neurotransmitters. On the other side, it may interact with other proteins, or it may simply be an ion channel that opens upon neurotransmitter binding. This is an electron micrograph of a synapse. This may look a little bit abstract because it is a transection of the synapse. However, it is still easy to see some of the key characteristics here. For example, we see mitochondria. There are lots of mitochondria here at the axon terminal. We also see active zones. These are black. If we zoom in and take a close look at this, we can see the synaptic cleft, which is not very clear but still exists on this graph. We can take a closer look at the distinctions between synaptic vesicles and dense core vesicles. Synaptic vesicles look like this. They're light in color, while dense core vesicles, which are another name for secretory granules, are black like this one. Chemical synapses can be classified in two ways. Firstly, we can classify them by presynaptic and postsynaptic component. The presynaptic component is usually always the axon. The postsynaptic component can vary. If the postsynaptic component is the dendrite of another neuron, then we call it an axodendritic synapse. If the axon contacts the soma of another neuron, then it is an axosomatic synapse. The axon of the presynaptic neuron can even contact the axon of another neuron. This type of synapse is called axonic synapse. The second type of classification is by symmetry, or we should say by asymmetry. Grace type 1 synapses are asymmetrical. The postsynaptic density is much larger than the presynaptic active zone. This type of synapse is usually excitatory, meaning that an action potential in the presynaptic neuron will depolarize the postsynaptic neuron, making it more excitable. On the other hand, synapses with symmetrical membrane differentiations are called Grace type 2 synapses. They are usually inhibitory, meaning that an action potential in the presynaptic neuron will usually inhibit or hyperpolarize the postsynaptic neuron, making it less excitable. Now, please take some time to pause and review. Can you name 2 organelles that are present in the presynaptic portion of the synapse? Well, we can find mitochondria, which are used to provide energy, and of course synaptic vesicles, which is the easiest form of organelle. The neuromuscular junction is a prototypical chemical synapse. It is the very first electrical synapse that was studied because it is large. It is the very first type of chemical synapse that was studied because of the fact that it is so large. The axon branches many times, as seen in this figure, and contacts the muscle at multiple points. This allows one single neuron to innervate a whole muscle cell and cause the whole muscle cell to contract. Let's zoom in at the details. First, this is the axon of the presynaptic neuron. Then, this is the neuromuscular junction, abbreviated as the NMJ. In the presynaptic component, we see lots of synaptic vesicles that are arranged in lines. Contained within it is a kind of neurotransmitter called acetylcholine. Acetylcholine is a small molecule neurotransmitter. It is then released into the synaptic cleft. One peculiar thing about neuromuscular junctions is that there are little grooves on the postsynaptic cell which is this time a muscle cell. These folds are called junctional folds. These junctional folds allow acetylcholine to stay within the groove for a relatively long period of time, thus completely stimulating the muscle to contract. This makes sure that one single neuron can make a whole muscle cell contract. Later, we will use the neuromuscular junction many times in the analysis of how the synapse works. Neurotransmitter synthesis. Of course, if we want to use neurotransmitters, they must be synthesized in some way. There are 2 major types of neurotransmitters. They are neuropeptides and small molecule neurotransmitters. They are synthesized in different ways. Neuropeptides are usually synthesized at home, or in other words, at the soma, just like any other protein. Usually, a propeptide is synthesized at first. This peptide is not the exact neurotransmitter that we will use in the synapse. This precursor peptide, or propeptide, is then processed in the Golgi apparatus. Then it is pinched off into a vesicle and transported along the axon until it reaches the synapse as a secretory granule. On the other hand, small molecule transmitters are usually synthesized on site. This means that there exist synthesizing enzymes located at the synapse, and neurotransmitter molecules are synthesized by them. The synthesizing enzymes themselves are made at the soma, just like neuropeptides. And we also need to transport these enzymes to the synapse in order that the neurotransmitter can be synthesized on site. The process of transport requires axonic transport. This process of transport is called axonic transportation because it is a kind of transport along the axon. We see 2 types of axonic transport. One is anterograde transport. Anterograde means towards the front. Anterograde means along the axon to the axon terminal. This type of transport uses the kinesin molecule. The kinesin molecule is the molecule you see in this figure. On one side, it holds up the vesicle using 2 hand-like structures. On the other hand, it uses 2 feet-like structures to walk along the microtubule. In this process, the kinesin uses ATP. Firstly, its 2 feet are all on the microtubule. Then ATP is turned into ADP, and then 1 feet is freed to move forward. And then this process is repeated, and the other feet moves forward, just like walking. And the kinesin walks to the end of the axon terminal. On the other hand, we also have retrograde transport. This is in the exact opposite direction of anterograde transport. The vesicles are carried from the axon terminal towards the soma. This type of transport uses a protein called dynein, which is very similar to kinesin and also walks on 2 feet. Retrograde transport is not very common. However, some viruses make use of this kind of transport. For example, the rabies virus uses this type of retrograde transport. If someone is bitten by a mad dog, then the rabies virus will follow the axon and go along into the spinal cord, where it wreaks havoc, and in the end disrupts the body function. Now that the neurotransmitters have been synthesized, let's take a look at how they are secreted into the synaptic cleft. This process is called excitation-secretion coupling. The excitation is the action potential. As the action potential reaches the axon terminal, it triggers the secretion of neurotransmitters. So these two processes are coupled. How are they coupled? Well, firstly, secretion is realized through exocytosis. The vesicles open up to the outer side of the cell and release its neurotransmitter content into the synaptic cleft, to the exocellular space. Vesicles are usually encapsulated by clathrin molecules. This clathrin molecule is boomerang-shaped. Many clathrin molecules can form a cage around the vesicle, maintaining its spherical shape. When exocytosis is performed, the clathrin cage opens up a little bit to allow the vesicle to fuse with the cytoplasmic membrane. Another set of very important proteins are called SNARE proteins. SNARE is a very figurative name. The proteins capture the vesicle on the cytoplasmic membrane just like what a snare does to a prey. However, SNARE is actually an abbreviation. SNARE stands for SNAP receptor. SNAP in turn means soluble NSF activating protein, while NSF is a chemical called N-ethylmalamide sensitive factor. The function of SNARE proteins are disrupted by botulinum toxins. If you put your food in the air for too long, then sometimes a bacteria called botulinum will breed on your food and produces botulinum toxins. These toxins are very toxic and can be deadly since they disrupt SNARE proteins and prevent synaptic transmission, thus paralyzing your muscles. The process of SNARE proteins is in short like this. Firstly, the vesicle binds to the SNARE proteins on the cytoplasmic membrane. Then, calcium ions enter the cell, and finally, due to the triggering of calcium ions, the vesicle fuses with the cytoplasmic membrane and releases its content. Let's take a look in more detail. There are two types of SNARE proteins, V snares and T snares. V stands for vesicle. V snares are proteins on the vesicular side. These include VAMP, also called synaptobrevin, and synaptotagmin. On the other hand, we have T snares. T snares include syntaxin and SNAP25. The process of triggering is like this. Firstly, a factor called NSEC1 is removed from syntaxin and SNAP25. Then, VAMP binds to syntaxin and SNAP25 and forms a trimeric complex like this. Then, calcium ions enter the cell and it helps phosphorylate synaptotagmin. And finally, a series of conformation changes pulls open the vesicle, causing it to fuse with the plasmic membrane and releases its content into the extracellular space. Now let's pause a little bit and take a look at the process of this exocytosis. What triggers the exocytosis? Well, it is actually the calcium-dependent phosphorylation of synaptotagmin. And where do the calcium ions come from? Well, then we have to go back to the action potential. When the action potential reaches the axon terminal, the cellular potential of the axon terminal temporarily goes up. The axon terminal is depolarized, and the membrane potential reaches positive values. There are many voltage-gated calcium channels located on the membrane of the axon terminal. Upon the activation by the action potential, the calcium channels open and allow huge amounts of calcium to flow in very rapidly. This is due to the large number of calcium-gated channels. This is also related to the huge concentration difference between intracellular and extracellular calcium ions. As we have seen before, the calcium ions outside the cell only have about 2 micromoles of concentration, while inside the cell, there is only 0.0002 micromoles of concentration. This concentration gradient makes sure that calcium ions flow in very rapidly in order to trigger neurotransmitter release. This quick inflow of calcium ions makes sure that neurotransmitter release is very timely. There are two types of exocytosis. One is more economical. We can see it on the right side of this figure. This is called reversible exocytosis. The vesicle does not completely fuse with the membrane. Rather, it only opens a little hole and then goes back into a full vesicle. This process is also called kiss and run because the vesicle only kisses the membrane temporarily and then runs away. On the other hand, we have the irreversible process, which uses the endosome system. The vesicle fuses completely with the cytoplasmic membrane, then a new vesicle is pinched off, which fuses with the endosome and later becomes a usable synaptic vesicle and is filled again with neurotransmitters. These two processes are both used in the axon terminal. Another very important characteristic of synaptic transmission is quantal release. This refers to the fact that neurotransmitters are always released in quantums. That is to say, there is no slide bar by which we can control the amount of neurotransmitters however we want. We cannot release 1 micromole, 2 micromole, 3 micromole, but rather we have a larger unit called a quantum. We can only release 1 quantum, 2 quantum, 3 quantums of neurotransmitters. Each quantum is equal to the neurotransmitter content of 1 single synaptic vesicle. We have 2 evidences for this. Firstly, sometimes at the neuromuscular junction, there is spontaneous release of neurotransmitters. This release causes a very small depolarization in the postsynaptic cell. This potential that caused in the postsynaptic cell is called the miniature end plate potential. End plate is just a special term to refer to the muscle cell that is postsynaptic in the neuromuscular junction. The miniature end plate potential is very small. In fact, the miniature end plate potential is the content of 1 single vesicle. The neurotransmitters contained in this 1 single vesicle usually opens about 1,000 receptor ion channels in the postsynaptic end plate. Then it causes a miniature end plate potential of around 0.5 microvolts. This value is usually always fixed. It does not vary at one synapse. Therefore, we know that this might be the basic unit of neurotransmitter release. Another evidence is evoked end plate potential. If we artificially give the presynaptic neuron a stimulus, then we can evoke an end plate potential in the postsynaptic muscle cell. The evoked end plate potential is usually always integer multiples of the miniature end plate potential. If the evoked end plate potential is 1 times MEPP, then 1 vesicle is released. If it is 2 times, then 2 vesicles are released. If it is 3 times, then 3 vesicles are released. This shows that neurotransmitter release is also measured using quantums, using the number of vesicles, rather than using a specific amount of matter. In this episode, we have seen electrical synapses and chemical synapses, and we have explored a lot the presynaptic component of chemical synapses, including neurotransmitter synthesis and excitation-secretion coupling. We have also met the neuromuscular junction, which we will continue to meet in the next part of this episode. In the next part, we will focus on the postsynaptic component and also talk about synaptic plasticity. That's all for this part of episode 5. Thank you for watching. See you in the next part.